with 100 participants at the moment, we have received way more interest than we thought when we started this endeavor. And in this introduction, you can see the banner up there that you have familiarized already with. It was on the website. And you see the title of this little presentation. I'm going to show you what really matters is what's coming ahead. But I thought I will try to set the mood for what we are going to hear about these days. And the presentation is entitled, What is so hard about the human thalamus? Why it matters and how to find out. I'm Giulio Pergola. I'm connecting from Bari, Italy. And the idea that this is hard as a topic comes from my own experience. You probably have all shared some of these experiences. It's happened to me, for example, with 2 papers and with 1 grant, that people cannot easily understand what's going on about the human thalamus, to such extent that this couple of reviewers wrote me something like, well, you know, this is complicated. This is going on with the thalamus in your data and in your grant. Why don't you take it out? Let's just focus on the cortex. Let's just focus on the hippocampus. It seems to me like the thalamus doesn't make for a very good advertisement. Why is that? Why is that so? I think one reason is it's somewhat complicated. And let's see why it is so hard and why it matters. So when I was a PhD student in Bochum, Germany, I invited Andrew Mays for a symposium, and his opening slide had this had this text that was fixed instantly in my memory. It was, if research on the hippocampus is hard, research on the thalamus is off the scale, which, as you can understand, is not what probably motivates a PhD student the most, but you know, we are also motivated by challenges. I remember that because he was very committed to research on the hippocampus, and he recognized that there was something hard about the thalamus. I think this hardship stems from the very structure and architecture of the human thalamus and how it is peculiar and how unique it is in terms of anatomy and function. And for my PhD, the main inspiration and the top paper that I was looking up to was the one you all know by John Eggleton and Malcolm Brown. And we are very fortunate to have John Eggleton here today to give the first talk. And I think the underlying idea that made that proposal John Eggleton and Malcolm Brown made so successful was that it captured the essence of the uniqueness about the thalamus, and it was that they hypothesized that there were segregated pathways subserving different functions. And so the proposal was that the anterior nuclei were in one pathway and the mediodorsal thalamic nucleus was in another pathway, and they were differently involved in different memory functions. And I think today, 20+ years afterwards, this idea has been accepted. This is coming from a paper by Michael Alassa, who will also join us and give a talk tomorrow. And the idea is that we have different pathways. They are not touching each other. The nuclei are not that much connected to each other, as it happens, for example, in the basal ganglia. And that's a fundamental principle that regulates the role the thalamus has in the brain. And the thalamus is highly conserved across species. It's not something that is coming anew for primates or for mammals. It's highly conserved. The question is how we can understand it. And there's a lot of different models that have been put forward to try and explain what's peculiar about the thalamus. Many models trying to understand a unitary vision of how the thalamus works across species. And my idea is to take a metaphor to explain how challenging it is to work with the thalamus. And the metaphor is that of a master switchboard. A switchboard has many different pathways. It's a place you might have in your house. If you have renovated your house, you have probably crashed against this issue of understanding what is going where and how do I light up, how do I switch on a light in that room, and how do I control that when my fridge is on, not necessarily the rest of the house has to be on. And if you have renovated your house, you might think that it's very critical to have a clear idea of where these wires are going and which label each of these switches has. The problem, and that's a big but that we have with the thalamus, the but is we don't have the labels. We're trying to define these labels. What is the function of this nucleus or this other nucleus? What is switched on by that nucleus? And the switch in the case of the thalamus is particularly poignant because, as you know, thalamic cells have a two-way mode of function. They can be on, they can be off, and they transmit information in a way that is depending on that state. But we don't have the labels. And so we don't know that the air conditioning is in one spot and the fridge is in another and the lights are in another. And the second but is that the wiring, the most accurate wiring maps that we have are really coming from another house, and that's rodent studies. So we have animal models that are accurate as far as the wiring is concerned, but when we try to apply that to human function and to human thalamic architecture, it becomes all somewhat fuzzy, like in this little image I put up there. And so we are trying in many different ways in animal models and in neuroimaging to put some labels there. The idea proposed by Eggleton and Brown was that two nuclei might subserve different functions in memory, and you can extend this to multiple nuclei. So we published this meta-analytic paper last year about the roles of different functions of different nuclei as assessed by functional magnetic resonance imaging. We have the summary figure here. The left thalamus is here, the right thalamus is here. And what we tried to do was to parse the activations found in all papers we could find that use a task-based fMRI to assess cognitive function. We parsed them into a dozen different domains. And then in the end, we colored what was activated in each domain, and we separated the different nuclei. Of course, it's a relatively coarse resolution. It's a very coarse resolution, I should say, because it's a meta-analysis of functional magnetic resonance imaging data. But the answer we got was not very clear. We had 93 studies. We had almost 2,000 participants. We had a good agreement about what the task was assessing in the experiment. But still, you can see that thalamic nuclei in humans appear quite promiscuous, at least at this resolution. And that's the reason why we probably need better techniques to improve this resolution. This is one issue. The other issue I have with this is that probably we are looking at categorizations that do not exactly fit the sort of operations that thalamic nuclei are doing. And so one direction is to think about different models that can help us put better labels on these switches. I think it really matters to understand this. On the one hand, because each brain that we know needs a working thalamus. As I said, it's highly conserved. It means it's important for the function of the entire brain. And I think we have evidence that tells us that this is important. Look at the red square at the center of the slide. That red square represents the average size of focal thalamic lesions. And the white rectangle that you see, that is the entire slide, represents the size of the brain. To have patients who manifest significant deficits in cognitive skills, you just need that little spot. If this symposium were about the basal ganglia, we will probably not get that. In fact, in the basal ganglia, as people age, there's an accumulation of very small lacunae that are brought by microinfarcts, and you can measure a gray matter decrease, which is sizable in comparison with focal thalamic lesion, but it does not impact function in the way a thalamic lesion can. This is to say that clinical evidence tells us that these switches, these little pathways that accumulate all in the center of the brain, in the thalamus, seem to play a role in human cognitive function. And the second reason why I think the field is realizing slowly that it matters to study the thalamus is that there's a raw study I have shown, and I just collected a few, the few that I really could come up with. And they're showing that the thalamus, when you measure it via neuroimaging, predicts a lot of stuff. This has come just 2 weeks ago. It's been published 2 weeks ago in Molecular Psychiatry, and it's entitled "A Thalamus-Centric Neural Signature for Restructuring Negative Self-Beliefs." It's just one piece of evidence that is continuously accumulating, but there's really a lot. People can predict psychiatric disorders, cognitive deficits in neurological patients. The activity of the thalamus can predict cognitive functions in animal models, in living humans, in epilepsy patients, and so on. And so this is the second reason why it's very important from a neuroscience perspective to study how the thalamus predicts these outcomes. The question about how to find out is what united all the organizers of this symposium. How can we find out with better methods, with better concepts and ideas, the importance of these switches, or I should maybe call them bottlenecks in these networks that help us measure stuff that we're interested in. As a group, it's been a great group effort, and I'm really happy to be part of this team. You will get to know the entire team in these 3 days. As a group, we focused on 3 aspects. One was the image processing aspect of the human thalamus, because of course, with animal models, you do not need the same sort of techniques to look inside the brain. You can use more accurate techniques. And so this will be the focus of the first day. The second will be about basic neuroscience. What is the unitary function of thalamic nuclei, and what is the particular function of individual thalamic nuclei, and how all we understand about this issue can help us, for example, understand inter-individual variability in cognitive measures and in clinical measures. And the third day is dedicated to clinical applications. The thalamus is involved in a lot of different clinical conditions, and we just have a small sample here of excellent speakers who will guide us into the latest findings about the thalamus. I'm very happy to be here, and thank you for being here. And I will now pass the word to Michael Hornberger for the introduction of our first speaker today. Thank you all.